Good morning, good afternoon, or good evening. This brings us to Chapter 5, and we are still on the topic of physiology and biochemistry of the skin. And now, today, we're going to talk a little bit about the dermis and the dermal-epidermal junction. So, in Chapter 4, we talked about the epidermis, and now we're going to go a little bit deeper. The dermal-epidermal junction is the barrier between the dermis and the epidermis. So, in talking about the dermal-epidermal junction, we'll briefly mention hemidesmosomes, which are very important. If you remember, we talked about desmosomes as the sort of morphological components in the epidermis that connected the keratinocytes together. You'll see here that hemidesmosomes, they're called hemidesmosomes because they don't connect two keratinocytes, but they actually connect a keratinocyte from the stratum basal layer to the dermal-epidermal junction. In talking about the dermal-epidermal junction, there's a section known as the basement membrane, and we can break that down into two different parts. One is called the lamina lucida, the other is the lamina densa, and then we'll also talk about anchoring fibrils. So these four components, hemidesmosomes, basement membrane, and anchoring fibrils, these all make up the dermal-epidermal junction. The dermis is primarily composed of two distinct morphological components. One is known as the papillary dermis, and the papillary dermis is the part of the dermis that is closest to the dermal-epidermal junction. And then we have the reticular dermis, which is the deepest part of the dermis. We'll also talk about connective tissue and epidermal appendages, which are, as you remember, the pilosebaceous unit, the nail unit, the sudoriferous glands, and things like of that nature. And we also will discuss the cells of the dermis, primarily in their context to both the fabrication of collagen and elastin and its destruction, but also for the immune cells that are present in the dermis. So this is the same illustration that you had in the last chapter, showing you the different sections of the skin. Here you can see the epidermis is up here in the top, with the stratum corneum being the outermost layer, and then we have the dermis here directly below that. Again, the boundary between the dermis and the epidermis. This is known as the dermal-epidermal junction. Notice when you have a hair fiber present, this gets rather complicated, as you know, this is an invaginated structure into the dermis. That's why it's called an epidermal appendage. And below the dermis we have the subcutaneous tissue, which is also often referred to as the hypodermis. So the dermal-epidermal junction, I mentioned the four parts of the dermal-epidermal junction, and one of the parts is called the basement membrane zone, or actually it's just called the basement membrane. So not to confuse you here, but sometimes the entire dermal-epidermal junction is referred to as the basement membrane zone. It is essentially, as I said already, the interface between the dermis and the epidermis, and it provides, the dermal-epidermal junction provides support for the epidermis. And again, just breaking it down into the three morphological divisions, we have the hemidesmosome anchoring filament complex. This essentially, as I mentioned before, the hemidesmosomes bind keratinocytes to the basement membrane. You'll see an image of that in a moment. And then the basement membrane, again, we have the lamina lucida layer. This essentially contains different types of structural proteins. You can see it gets its name from the laminins. That's an important type of structural protein here. There's also intactin, ctogen, and fibronectin that are also present. And these are all non-collagen type structural proteins. When we look at the lamina densa, which is the layer below, as you can imagine, lucida and densa, these names come from descriptions that were given to them due to actually transmission electron microscopy studies. And lucida meant that when they did the studies, when they took images of this section of the skin, this part of it appeared clear for lucid. Densa referred to the part of the basement membrane that actually looked dark in the TEM images. So it's just something for you to know. Essentially with the TEM, they stain the sections of skin, and if they appear, the section that appears clear, the upper layer is called the lamina lucida, and the lamina densa, which is darker, is called lamina densa. So the lamina densa actually carries out a really important function as a barrier, and it actually filters larger molecules, and anything greater than 40 kilodaltons cannot pass through this zone. And also in the lamina densa, unlike the lamina lucida, which has non-collagen proteins, in the lamina densa we have type 4 collagen present, and that's really important for skin structure. And then again, this third morphological division, the anchoring fibrils, and you'll see an image of that shortly. So this is an image of the dermal-epidermal junction, and it's a nice illustration showing you how the different proteins interact with each other. And you know, in this section, you can see the breakdown of different molecules: nitogen, perlecan. I don't really make that much reference to these in the text. The important ones are laminin, very important, type 4 collagen. Also integrin is important as well. That's a key protein here. But you can see the integrin molecules are shown sort of jutting out from below. These are connected to the laminin structures. And then you can see that, you know, this is all sort of interweaved with the type 4 collagen, which is shown by this red type structure that's here, with a little sort of elliptical head. So I mentioned, how do we connect the epidermis with the dermal-epidermal junction? And this is a figure that shows how that occurs. Here you can see the lamina lucida, which we mentioned before, the lamina densa. So these lines are just shown here to make it so you know that it's like a darker section. Inside the lamina lucida, you can see there's these anchoring filaments, the laminin structures. And then, if you can imagine, right above this is just one cell that's sitting above here. This is one keratinocyte from the basal layer, and you're not really seeing the whole cell. You're only seeing like a small section of it, right where the plasma membrane would be. And what it's showing you is that there's a hemidesmosome here, and it's connected to the lamina lucida. And to the hemidesmosome, we have these intermediate filaments, which are also called tonofilaments. These are connected to that. So this is how the basal cell keratinocytes are anchored into the dermal-epidermal junction. And again, this up here is this is just part of one keratinocyte cell that's bound to the lumina or the lamina lucida. Down below, you can see the lamina densa. We have these anchoring fibrils, and these are actually type 7 collagen. These are connected to larger collagen structures such as type 1 and 3. And again, these are important in the dermis. You can see that you have these sort of anchoring plaques type structures. And you also have where these loop around and form these what are called anchoring fibrils. So if we break down everything in the dermis, we can sort of, just to get a general idea of all the component parts, in the papillary dermis, which is this upper part, you can see that we have collagen bundles that are mostly small diameter collagen. We have what are known as oxytalin elastic fibers. So if you might have heard of elastic fibers or elastin. Elastin is an important protein that helps the skin regain its shape if you stretch it or you deform it in any way. When it springs back to its original shape, this is due to elastin. We also have types 1, 3, and 6 collagen predominantly. There's also some 5, but predominantly 1, 3, and 6 collagen are predominant in the papillary dermis. We have ground substance, and this is sort of like a matrix type material that the elastin fibers and the collagen are embedded in, and we'll talk a little bit about the components of the ground substance later. We also have fibroblasts present here and mast cells. These are immune type cells. In the reticular dermis, we have large diameter collagen bundles. We also have another type of elastic fiber, which we'll discuss in a little bit. And then we have types 1, 3, and 6 collagen in the reticular dermis, and we also have ground substance. The dermis, again, contains fibrous material, and the fibrous material is the elastin fibers and the collagen fibers. It also contains filamentous components, which we haven't talked about yet. And then it has the amorphous connective tissue, and the amorphous component is the ground substance that we were talking about. We also have nerve networks that are hosted here. We have a much more extensive nerve network in the dermis than we do in the epidermis. We have vascular networks, so we have arteries coming to this section of the skin. We also have the epidermally derived appendages, so teeth, nails. We mentioned pilosebaceous structures and sudoriferous glands already. We have the fibroblasts, and again, these are the cells that are responsible for the synthesis of collagen and elastin, and they're also responsible for the breakdown of collagen and elastin in some cases. We have different types of immune cells that are present. First, the macrophages. We also have mast cells, and then we have other bloodborne cells such as lymphocytes, plasma cells, and other leukocytes. So in general, the dermis provides the skin with pliability. It also provides it with tensile strength. So it's a strong material if you try to pull it apart. If you have like a strip of skin and you try to pull it from both ends, it's a fairly strong material. And a lot of that is thanks to collagen. And the elasticity, as we already mentioned, due to the elastin that's present there. It functions by protecting the body from mechanical injury. It also is responsible for binding water, and it aids in thermal regulation. Part of this is through the blood. You can thermally control temperature by regulating the blood flow to a certain region, but also the sudoriferous glands, and that's going to be a really important section of the skin that we're going to actually cover in the next chapter, chapter 6. And we also know that the skin contains receptors of sensory stimuli, and again, that's the extensive nerve network of the skin. So when we talk about the connective tissue of the dermis, we can talk about collagen and elastin. That's the fibrous component. And then if we talk about filamentous glycoproteins, and also proteoglycans and glycosaminoglycans, these are both the diffuse and filamentous dermal matrix. So, essentially we have the fibrous proteins that are embedded in a dermal matrix that is composed of diffuse and filamentous components. And hopefully you'll see what I'm referring to in a few minutes. So collagen is the major dermal constituent, and it's essentially 75% of the dry weight of the skin. And again, it predominantly provides the skin with tensile strength, but also with some elasticity. I mentioned before the type 6 was one of the predominant. I meant to say type 5. Types 1, 3, and 5 collagen are the major collagens that are found in the dermis. Type 1, you can see it's really the predominant factor, accounting for 80 to 90% of the collagen. Type 3 is only about 8 to 12%, and type 5 is less than 5%. And if you remember from one of our earlier chapters on protein structure, you'll recall that the primary structure, which is the amino acid sequence of collagen, is essentially glycine and then XY, where X is equal to proline and Y is equal to hydroxyproline. And we also had this in one of our previous chapters where we showed the structure of the alpha chain in collagen. And again, please don't confuse this with an alpha helix. This is called an alpha chain. It's a different conformation. And here you can see the structure of this alpha chain in A here. And then this ribbon kind of shows you how it wraps around. In B, we have a space fill model of one alpha chain. So this is it's called a van der Waals model. Essentially, it's the same as this. The only difference is that, all of the space is occupied in this by the atoms. So it's a more accurate description of the actual space that would be occupied by all of the atoms in the molecule. Here we have 3 alpha chains, which are essentially wrapped together to form this super complex. And then, this part in D is just showing you if you rotate the 3 alpha chains toward you, you can look down the center here and you can sort of see this space in here. But this is like a ball and stick model, which is like A. But if you did a van der Waals model, like B or C, you wouldn't really see that very easily. Also note that the glycine is shown in red in D, so you can see that it's the internal part of the structure. We also showed this in our protein chapter, showing, I believe this is an SEM, or it's actually this might be a TEM for transmission electron microscopy or micrograph. It's essentially an image collected from a transition electron microscope, and you can see, this is a collagen fiber here, this is another one here, this is another here, and another here. And what you can see in these is that they have these little striations that are present, and these striations are essentially due to where you see, these gap type structures that occur in the collagen complex. So when you have, each one of these little sections here, this is a triple helix or 3 alpha chains wrapped together. You can see that you have like this section here where there's some space. Then you have another alpha chain down here, another one over here. And depending on the pattern that these present, you essentially have these bands that occur, and that's again essentially due to the staggering of these alpha chains, or of the 3 alpha chains wrapped together. In collagen, if you'll recall, it's cross-linked, and it's cross-linked. There's two predominant species responsible for cross-linking it. One is pyridinoline, which is the structure shown on the left, and the other one is glycoaldehyde pyridine, which is the structure shown on the right. What you'll notice is that both structures have this sort of aromatic type structure in the center with a nitrogen atom, and then they also both have these acid groups, carboxylic acid groups, that are on the end, and these are responsible for attaching at cross-linking points. And you also have amine functions as well. So these are both reactive points where you can attach the molecule to proteins. And that brings us to elastic connective tissue. Again, this is elastin. So we have essentially a continuous network from the lamina densa to the connective tissue of the hypodermis. And, as we mentioned already, we know that elastic fibers return the skin to its original position after stretching or deformation. So, as its name implies, elastin, it essentially gives the skin its elastic properties. We also find elastic fibers in blood vessels and lymphatic tissues, and also as part of the hair follicle structure. Actually, in the sheath essentially refers to the coatings of the hair follicle structure. We'll discuss that in another chapter when we talk about hair biology. But essentially, they're just different layers that surround the hair follicle, and collagen's there stabilizing that as well. Essentially, elastic tissue accounts for about 4% of the dry weight of the dermis, so it's much less than collagen. And the interesting thing about elastic fibers is they have a microfibrillar part, so they have like a fiber part, but they also have an amorphous component to them. And here we have this molecule called fibrillin. This is essentially a glycoprotein of elastic fibers. And the last thing I'd like to mention here is, again, when we break down the dermis into the two principal components, the papillary dermis and the reticular dermis, we find that in the papillary dermis we have oxytocin fibers, and in the reticular dermis we have elastin fibers. So in the papillary dermis, again, this is the dermis that's in contact with the dermal-epidermal junction. We have collagen fibers that have small diameter, or at least the bundles of them. Okay, and we have the oxytocin elastic fibers that we just mentioned, and essentially these are mature elastic fibers are not found here. So again, a lot of the elastic properties of the dermis, you'll find this is due to the reticular dermis. If you look at photodamaged skin, you'll notice that the elastic fibers are essentially more dense, large, and they have an abnormal shape. This is essentially damage to the elastic fibers. So if you look at anyone that has photodamaged skin, it's not very elastic. It doesn't spring back to its original shape, and this is essentially due to the formation of what's known as an elastotic material, and essentially it's damaged elastin. So the papillary dermis is actually designed to take impact, and one of the things, and this is mentioned down here below, is that part of this is due to the dermal papillae. And what these are is that if you remember looking at the figure of the skin structure, you'll notice that the border between the epidermis and the dermis, it's not a flat border. actually it's only flat in mostly in photodamaged skin, but in normal healthy skin, you actually have these sort of, it has like a finger-like structure or finger-like projections of the papillary dermis into the epidermis. So just to go back to recall for 1 second, this is this structure that you can see here. This is like the finger-like projections that you see that are shown here, and this increases the surface area of contact between the epidermis and the dermal-epidermal junction and the dermis. So that makes it stronger. So if you have like photodamaged skin, for example, and this becomes flattened, it becomes much easier to remove the epidermis from the dermal-epidermal junction or the dermal-epidermal junction from the dermis. And these interdigitate with what are known as reed pegs from the epidermis, so that it's just kind of like a connection point that they refer to them as reed pegs. We really won't get into that detail for this class, but we should also mention that in the papillary dermis we have a high density of fibroblast cells, and these are rapidly proliferating cells that obviously they have a high rate of metabolic activity, and we think of collagen and even elastin to some extent, but we also have other species that are synthesized by the fibroblasts, such as proteoglycans. And specifically you see that different species of proteoglycans are made by fibroblasts in the papillary dermis than there are in the reticular dermis. So if you'll notice on the slide of the dermis that we showed, we didn't really list fibroblasts in the reticular dermis, and that's not really correct. Actually, there are fibroblasts there, but not as high quantity of fibroblasts that you have in the papillary dermis. So in the reticular dermis, two major characteristics of it are you have large diameter collagen fibers, and you also have the mature elastic fibers. So these are the Elastin elastic fibers, and essentially these mature elastic fibers, they support and form a superstructure around the collagen fibers. So generally speaking, in the dermis now, we have the diffuse and filamentous dermal matrix, and again the ground substance. This is essentially one key component of the matrix. We have 0.2% of the dry weight of the dermis. Doesn't sound like very much, but it's really omnipresent in the dermis. It's all over the place, and it essentially surrounds and embeds all the fibrous components. And you'll see a nice structure of it, but it contains proteoglycans and glycosaminoglycans, and essentially these are responsible for binding water, and they help maintain moisture in the skin. The proteoglycans, there's three principal types that are present in the dermis. We have the chondroitin sulfate keratin sulfate pair, so they come in pairs essentially. You'll see what I mean when I show you the illustration of the molecule. You also have heparin sulfate proteoglycans, and we also have chondroitin 6 sulfate proteoglycans. So if we look at the proteoglycan structure, and this is a general structure, essentially the core, it's a huge molecule, and the core of it is hyaluronate. This is also hyaluronic acid, and it's shown here with this dark line that's sort of the backbone here. And off of that we have these sort of arms coming off. So this central piece here, this is actually a protein core molecule. So we have we have a polysaccharide, really long polysaccharide that's this hyaluronic acid. Hyaluronate just refers to if if you have an acid in biochemistry, oftentimes, at neutral pH, the acid will not have its proton, so you'll have COO minus instead of COOH. So we refer to it as 8 at the end, but I'll refer to them interchangeably. So hyaluronate or hyaluronic acid is essentially this polysaccharide structure that's formed at the backbone here. We have the protein core. So each one of these, this is a protein core coming down. And then off of the protein core, we can have either one or two types of structures. And these are essentially the proteoglycans that you saw on the previous slide. So in this example, if we blow this up a little bit or magnify it, you can see that, these longer red things here belong to chondroitin sulfate, and these shorter ones belong to keratin sulfate. And again, this is just an example of, the one type of proteoglycan. You could also have heparin sulfate present in that section, or you could just have chondroitin 6 sulfate. So again, hyaluronic acid is really important for binding water, or essentially the proteoglycans are really important, but hyaluronic acid is used very commonly in skincare formulations due to its moisturizing properties. So the cells of the dermis, we already mentioned the fibroblast, and again, these are responsible for the synthesis and degradation of fibrous and non-fibrous connective tissue. Another important cell group that we have, or type of cell, are macrophages. Essentially, these are immune system cells. They're phagocytic, which means that they can engulf and digest other types of organelles and even cells to some extent, and other types of foreign bodies that may present themselves in the skin. They're an antigen presenting cell, which means that they can sort of cleave a small peptide portion off of a foreign pathogen. It's like a virus, or even maybe off of a bacterial cell, and then they can present that antigen to lymphocytes of the immune system. And by doing that, that sort of launches an immune system attack, and that antigen that it collects, that fragment of peptide, is used by the lymphocytes to synthesize antibodies, which then fight against and neutralize the invading pathogen. So they're also microbicidial, and essentially they produce lysozyme, which can digest things, foreign bodies. Also peroxide and superoxide. These are free radical species, and they actually can be used to kill off foreign invaders by immune cells. The macrophages are also secretory cells. So we mentioned before growth factors and cytokines. Those are also secreted by keratinocyte cells, but that's a key role of macrophages as well. They're hematopoietic. It's very difficult to pronounce, as you can see, and essentially that just means that they give rise to red blood cells, and they're also tumoricidal in a sense. And macrophages are also very important for wound healing. We also have mast cells, and again, these are like a specialized secretory cell, and you find the greatest density of them in the papillary dermis, so right next to the dermal-epidermal junction, and you also find them surrounding the epidermal appendages. And likewise, you will find them around blood vessels and nerve fibers as well. So, one of the mast cells sort of are infamous for the role that they play in allergies. So, essentially they produce histamine, which triggers the inflammatory response. And that's what happens when you have allergies and things. These cells are activated and they produce histamine, and that's why a lot of the medications for allergies are antihistamine drugs. And in general, we see that the mast cells are very important in wound healing, and also again in just basic defense against pathogens. So that pretty much closes this chapter, covering the dermal-epidermal junction and the dermis. And just if you want to read up on any of this material in further detail, you can find most of that in chapter 1 of your book. So thank you for your attention, and we'll discuss some new exciting topics in the next chapter.